where we left off last time was talking about the role that ethylene plays in senescence and abscission. And the senescence aspect of ethylene, there was a lot of circumstantial evidence early on that ethylene played a role in senescence, but now it's pretty clear that it probably doesn't. The main role that ethylene is playing is associated with abscission. And when we think about abscission, we need to think about not just leaves, but also need to think about flowers. When flowers have been around a long enough time and have not been pollinated, the flower falls off. There's senescence in the petals and the flower falls off. Right? When fruit has developed to a certain state, it falls off. All of these involve abscission, breaking of specific tissues to shed that organ from the plant. And in the case of leaf senescence, right at the base of the petiole here, there's a layer called the abscission layer. And the thing that distinguishes that abscission layer from neighboring cells is its response to ethylene. Okay? So ethylene is produced locally in the region of the abscission zone. But the sensitivity to that ethylene changes as the amount of auxin from the leaf decreases as the leaf senesces. So leaf senescence induces a decrease in auxin, which increases the sensitivity of the abscission zone to ethylene. The result of ethylene in that abscission zone is twofold. First of all, there is the production of cell wall degrading enzymes. So in these specific cells, in this layer, cell wall degrading enzymes are produced. But it turns out that in most cases that is not sufficient to cause abscission. There's one other thing that is required, and that is there has to be sufficient water potential in the leaves. That is, when you degrade the cell wall, what's going to happen to the size of cells? When you degrade the cell wall, what's going to happen to the volume of the protoplast? It's going to expand, right? Because you're removing the thing that's constraining it. So the second part of this is the cell expansion. The cell expansion, it used to be thought that ethylene actually caused cell expansion, but there doesn't seem to be any evidence that supports it now. The cell expansion is a consequence of the cell wall degradation. You lose the constraint on the volume of the cell and it expands. And that expansion turns out to be important because the expansion is what actually breaks the vascular strands. Right? So the cells, as they get bigger, they push that abscission zone, they push the two halves of it apart. And that's what breaks the vascular strands and the leaf falls off. Okay? So the role that ethylene is playing in this has relatively little, if anything, to do with senescence, but everything to do with abscission. And what happens in flowers and what happens in fruits is basically the same thing. The ethylene is controlled in different ways, but the ethylene sensitivity of the tissues is the same. Okay? All right. One other thing that ethylene plays a role in that tends to be confusing for students is the maintenance of the hypocotyl or the epicotyl hook, depending upon where the cotyledon leaves are located. All right, so what's the role of the hook? Why is the hook there at all?
Did you say it was like protecting the cotyledon?
Yeah, basically that's what it is. So remember what's happening is this thing is pushing through the soil. Normally, seedlings don't grow like this in the dark. Right? They grow in the soil in the dark. And that hook is there to basically provide protection. The shoot apical meristem is sitting down in that sort of crotch between the two cotyledons. And so the hook is actually pushing through the soil. Okay? So one of the things that ethylene does is ethylene causes thickening of the tissues in the region of the hook and also causes the asymmetric growth. So where's growth going to happen more rapidly? On the top or the bottom side? got to be more rapidly on the top side, right, to keep the hook there. What happens when the seedling sees the light? What happens to the hook, Jonathan? And what causes that opening? What's the signal for it to open? I mean, light is the signal, but what perceives the signal? We only got two choices in plants, right? What are the two choices? Phytochrome or blue light receptors. So which one is it for hook opening? Which is the main one for hook opening? It's phytochrome, yeah, right? So without knowing anything else, what would you think the signal transduction pathway is for hook opening, light-dependent hook opening? Emmy?
Asymmetric growth of the end, but on the bottom side.
Yeah, and what would induce that?
Like, you said the signal transduction pathway, right? So probably the phytochrome would cause the bottom cells to expand faster.
And what else must happen? What else would you expect to happen?
Beginning.
Well, if it was before that ethylene caused it to grow this way, then either ethylene is like with breast now, or
Yes.
the distribution is different.
Yeah, either the distribution of ethylene is different, and that's certainly the case. Think about a seedling pushing through the soil. The ethylene diffusion around the seedling is going to be much different than when it pushes out into the air. Right? So there's probably a greater loss of ethylene. But when phytochrome is activated, the synthesis of genes involved in ethylene biosynthesis stops. Okay, so ethylene synthesis in the hook also goes down. Okay, so this opening and closing of the hook is a physiological response that is governed by ethylene and by phytochrome, which is meant to maintain the hook as it's moving through the soil and open up the hook and get all the other light-dependent morphology changes to happen once the seedling reaches the top of the soil. Okay, so one of the things that when we're talking about ethylene responses early on, we talked about the triple response in seedlings. Thicker hypocotyls, shorter hypocotyls, and ageotropic growth were the three components of that triple response. And one of the things that people have looked at is what causes the shorter hypocotyl and thicker hypocotyl. If cells take on a different shape during growth, what must be dictating that different shape? Yeah, the cellulose microfibrils, right? Those are the things that dictate the way a plant cell responds. So it shouldn't be particularly surprising to you that one of the effects of ethylene associated with the triple response should have something to do with the orientation of the cellulose microfibrils and what determines their orientation. What determines how the cellulose microfibrils are oriented? Yeah, it's got to be something in the cytoskeleton, right? So interesting study has been done to look at, these represent cells that are growing in the absence of ethylene in the hypocotyl, and you see the sort of, around the circumference of the cell sort of orientation of the cytoskeleton. In the presence of ethylene, the cytoskeleton reorients along the long axis of the cell, so cells tend to get broader and don't grow as much along their length. Okay, so the expectation that changes in the orientation of the cellulose microfibrils in the cell wall and the cytoskeleton that underlies the plasma membrane that dictates how those cellulose microfibrils are oriented, that those must change in response to ethylene. So how this happens, what's the ethylene signal transduction pathway that starts with the perception of ethylene and ends up with the reorientation of these cellulose microfibrils and the cytoskeleton? Don't know what the answer to that is. But you should be able to at least consider the physiological significance of this. Okay, last thing I want to say something about is probably the most common role that ethylene plays in plants, and that's wound responses. We learned that the potential for every cell to produce ethylene is related to the ability of every cell to respond to a wound. One of the obvious outcomes associated with ethylene response in a wound is turning on cell division again, right? Because what happens in a wound response is the tissue wants to create new cells, often sort of non-differentiated callus sort of cells, to basically cover the wound, right? So that one of the things that has to be a main component of ethylene responses in wounding is the turning on cell division to basically cover up the wound. But we also saw the other things that happened associated with wound responses. Ethylene can trigger auxin synthesis to guide the formation of new vascular tissues if the vascular tissue has been damaged. So the wounding responses in reality are relatively complicated, even though they're common to all different cells. And it's sort of surprising that the textbook says virtually nothing about wounding responses. One of the things the textbook does talk about, but doesn't relate to wounding responses, is the role that ethylene has been found to play in plant defense responses. So there are a lot of responses to pathogens that have been found to involve ethylene in their signal transduction pathway. And one of the questions from an evolutionary perspective has been whether or not wounding responses and defense responses have some common origin. It wouldn't be surprising that they might have some common origin, and therefore that ethylene plays a common role in both of them. But we haven't dissected either of those pathways far enough to really see whether or not that common origin is there or not. Okay, that's it for ethylene. Any questions before we go on? Yes? Yep. I wouldn't be surprised if they did, but I don't know the answer to that. What would you expect would happen?
They would just go on.
So what was the compound you were talking about?
MCT.
And what does that do?
That blocks the receptors.
It's an ethylene response inhibitor, correct?
So it should just.
So you would expect then it to behave like an ethylene response mutant. In the presence or absence of ethylene, it would behave as if ethylene wasn't there. I don't know the answer to that, but I'm almost sure that that would be the outcome of it. Yes?
Just a general question, because so far on the chapters about the hormones,
Yep.
they talk about, oh, this mutant is a mutant of this gene. How do they induce the mutation in that specific gene?
How do they do that? So when they talk about mutations, where do those mutations come from?
So basically, they just after they apply some sort of chemical, they just don't direct mutation.
Sure, that's one way to do it. So EMS. What is it? I can't remember what it stands for though.
Just describe it.
It's not ethylene, but it's something that intercalates into the DNA and causes random mutations when the DNA is copied. It's a very common mutagen. I can't remember. My brain is drawing a blank what it stands for. What else, though? I mean, that's one way to create mutagenesis. What's another way to do it? Sure. So what would you have to do then if you wanted to? That's right. So if you knew what gene you wanted to alter, you could alter that gene by putting in antisense RNA. We also talked about microRNAs, right? And microRNAs can cause degradation of complementary messenger RNA. So there's a lot of different ways that you could knock out specific genes if you know that's the sequence of the gene you want to knock out. In Arabidopsis, there's an even easier way. Who's an Arabidopsis person here? Nobody. In Arabidopsis, if you know a protein you want to knock out, you go to one of the Arabidopsis collections and you look down, you find a protein and you order the seed, because there is an insertion mutation in every gene in Arabidopsis that they have in a collection. You can just buy the seeds, assuming that the seeds are viable. So you don't even have to do any work anymore. Well, you have to look it up and pay the money, but other than that, so you can get mutations in any protein you want, right? So it's very easy to do.
How did they make that in the first place?
There's lots of different ways that you can make those. So you could do it by EMS, right? You could use EMS and just induce random mutations and go through and sort it all out. But the easier way to do it is use transposons or something that does random insertions in the DNA that leaves a trace of where that insertion was, so that you can use molecular techniques to find out where that insertion has happened. Yeah, so someone goes through and generates tagged libraries of insertions in every gene, and that's available in Arabidopsis and several other plants now. Okay, so what's another way of doing random mutagenesis that we talked about just the other day when we talked about cytokinins? Agrobacterium. What does the tDNA do? Inserts. And how does it insert? Specifically or randomly? And if it inserts in the middle of a gene, is that gene function going to be lost? Probably, yeah. So tDNA tagging is another way to do it. So there's lots of different ways to get at these things. Most of it is done sort of with a forward genetics approach. A reverse genetics approach. Start with the gene and then do something and then see what the phenotype is. That's a more common approach these days. Okay? Other questions before we go on to abscisic acid? Okay. All right. So far we've talked about the role that hormones play in regulating growth. And abscisic acid was discovered because of a role it plays, a different role it plays in regulating growth. And that is that, well, we've talked about cells in plants, cell division is limited to relatively few places, right? It's limited typically to the meristems. But that's not always the case. There are some places where wounding, for example, where cell division turns on again. There are a couple times when cell division is intentionally turned off and then turned back on again, and that has to do with dormancy. What do we mean by dormancy? Grace? Well, so our parenchyma cells in the vascular tissue of a plant dormant? Would we call those dormant? They're not dividing, right? But there's something different about dormant.
They don't even metabolize. They're just like all cellular processes just stop.
That's often a component of what precedes dormancy, but it's still not quite right. What are some examples of tissues in plants that are dormant? Seeds, right? What else?
Lateral buds. Lateral buds.
Buds, yeah. And one of those buds, we can think of axillary buds as essentially being dormant, but also this time of year we're seeing buds that were formed at the end of the fall last year that stopped growing, and now with the spring conditions they're starting to grow again. But we still haven't answered the question, what do we mean by dormancy? It does have to do it has something to do with sort of growth starting up again.
Does it require an environmental signal?
It typically requires an environmental signal for it to happen. And so that's sort of the key component of it. Dormancy is a suppression of growth under conditions where you would expect it to be able to grow. So for example, seeds. In order for a seed to grow, we'll talk about what happens in seed development in just a minute, but the last stage of seed development is drying down, right? The suspension of metabolism that Hemi was talking about. And in order for the seed to germinate, one of the things that has to happen is the seed needs to rehydrate. It needs to take up water. It's called imbibition. Strange word. Imbibe. Taking up water. And some seeds, when they take up water, they germinate. You put them in the right conditions, right temperature, water availability, they imbibe, and the seeds germinate right away. Dormancy is when the seed takes up water but still doesn't grow. When the bud is under, it's got the environmental conditions, it's not the middle of winter anymore, springs come out and it still doesn't grow. Okay, so it represents a metabolic, a physiological constraint that prevents the cells from growing. And so obviously the role of the hormones that we're going to talk about is what is inducing this dormancy and what releases this dormancy. And obviously these are physiological traits that have evolved to, for example, allow tissues to survive the winter, buds to survive the winter, or seeds to be able to survive. Well, there's lotus seeds that survive for thousands of years, right? So to maintain themselves in some sort of metastasis where they're not growing, and then under the right conditions start growing again. So ABA was really discovered associated with trying to understand what controls and what releases dormancy. And this picture here shows you one of the early mutations that affects dormancy. So here's corn cob, and what we see is even before the corn seeds have dried down, they've germinated, right? So we see little seedlings coming out of these guys. It's called vivipary. Premature germination of seeds, often very early in the seed forming process. Right? So in other words, dormancy was never established in these seeds. The embryos were formed, and as soon as the embryos were formed, the seedlings started growing. Right? Nothing held back the growth of that embryo. In normal seed development, that embryo would be held back for some period of time until the dormancy was released. And this is where ABA came in. Interestingly, ABA was first purified as a compound that was involved in the abscission of cotton fruits. And that's how it got its name. But as we'll see, ABA typically plays no role in abscission. The big role of ABA is related to dormancy, and we'll also see that it's involved in a lot of different types of stress responses. One interesting thing that the textbook talks about that we'll come back to cover to a brief extent in a few minutes is the possibility that ABA is not unique to plants. We have ABA in us. And ABA plays a role in stress responses in lots of different types of organisms. Yeast, flatworms, and humans, as well as plants. Right? And one of the things that is interesting about this is there seems to be a common signal transduction pathway for all these stress responses in plants as well as in humans. And interestingly, that common signal transduction pathway involves G protein linked receptors. So remember back when we talked about signal transduction pathways? We said that plants don't have G protein linked receptors. Well, apparently they do. Whether they play a normal role in ABA responses or whether there's something very special we don't really know the answer to this. As we pointed out, we don't even know what the ABA receptors are yet. And we'll talk about that in just a minute. So there's lots of interesting questions about ABA from an evolutionary perspective as well as a physiological perspective that we just really don't know the answers to. Okay, so ABA is an interesting compound. You might, from looking at its structure, you might guess right away that it's a terpenoid. It's basically a hydrocarbon with alternating single and double bonds at various places. It's a 15-carbon terpenoid. And one of the things you may remember from when we were talking about terpenoid biosynthesis is that the 15 and 30 carbon terpenoids are made in the cytoplasm, and at the 10, 20, 40, and greater are made in the plastid. So you would suggest then from this that ABA is synthesized in the cytoplasm. But it turns out that the mutations that have the biggest effect on ABA biosynthesis occur in the chloroplast. And the reason for that is that the precursor of this 15-carbon compound is something we've already seen. Zeaxanthin, a carotenoid, a 40-carbon compound, is the precursor of ABA. So if we look at the biosynthetic pathway, it's in the plastid, and zeaxanthin is an intermediate in the synthesis of ABA. We've already talked about zeaxanthin in two different contexts. One, we talked about zeaxanthin and its role in photoprotection. That is, dissipating excess light energy in the antenna to try and limit light-induced damage in the photosynthetic machinery. We also talked about zeaxanthin as potentially being the blue light receptor for stomatal opening and closing, that in the guard cell chloroplast, zeaxanthin has been proposed to be the compound that actually absorbs the light and initiates a signal transduction pathway for light-dependent stomatal opening and closing. So now here's another role that zeaxanthin plays. It's a role that zeaxanthin plays as a precursor of ABA formation. And one of the things that has to happen, zeaxanthin has got 40 carbons in it. ABA has got 15 carbons in it. So somewhere there's a cleavage that happens in here that takes this 40 carbon thing and breaks it into a 15 and a 25 carbon thing. And we'll talk about that in just a minute. But I want to challenge you to think about something. Where is zeaxanthin located? It's in the chloroplast, but where in the chloroplast? Where do we talk about zeaxanthin's location? Nope, it's not in the stroma. Remember that one of the things it can do is it can take excited states from chlorophyll and dissipate it as heat. Right? It's getting rid of that light energy. So if the zeaxanthins are taking energy from the chlorophyll and converting it into heat, where are they located? Well, let's be more specific. They're not on the thylakoid membranes. Yeah, they're part of the photosystems. They're in the proteins that make up the photosystems. They're in the antenna proteins. And I apologize, I didn't put this in the slides until this morning because I realized that this is an important thing to be thinking about. So here is one of the main light harvesting proteins that is associated with photosystem 2. And in these light harvesting complex, there are typically 3 carotenoids. One of them is right here. You can't really, I couldn't find one with a different picture, or a different color. But here's one right here. There's another one right here. And the third one sits here like this. They are all embedded in the middle of proteins that are embedded in the thylakoid membrane. The purple stuff are pigments. So there's chlorophylls here. Can you really see any chlorophyll rings here? There's a chlorophyll ring right there. There's another one right there. So there are actually 7 chlorophylls and 3 carotenoids in this complex. And say that again? Oh, these are, the red are the alpha helices and the greens of the protein, and the greens are the loops of the protein that connect the alpha helices. I'm sorry, I should have pointed that out. But the thing that I really want you to see in this is that the carotenoids, the zeaxanthin that is present in here, is embedded in the middle of this protein, in the middle of the thylakoid membrane. So if we think about what's happening in the biosynthetic pathway, this conversion of zeaxanthin to violazanthin to neaxanthin to a cis form of neaxanthin and the cleavage are all happening in the thylakoid membrane. Zeaxanthin is not water soluble. It doesn't float around. It's stuck in the middle of this hydrophobic interior of the protein. So how do you get an enzyme in there to catalyze these reactions? We don't know. It certainly happens, but we have no idea how it can get in there and manipulate the characteristics, the chemical composition of these compounds when they're embedded in the middle of a protein. Interesting question.
Is there no production of chlorophylls afterwards? Because I'm sure the chlorophylls that aren't
Say that again.
Are there other chlorophylls
Are there chlorophylls
synthesized?
being synthesized?
Yeah. Outside the photosynthesis.
The chlorophylls, chlorophyll biosynthesis, up to a certain point, does occur on proteins that are sort of floating around free in the stroma. But once the chlorophyll becomes capable of absorbing light, once the intermediate becomes capable of absorbing light and creating excited states, every one of those intermediates after that is bound in a protein. Because if they weren't bound in a protein, when they absorb light, remember one of the things we said that can happen when a chlorophyll absorbs light is that it can sensitize the formation of singlet oxygen. Remember, chlorophyll in the presence of oxygen and light is the most dangerous biological molecule. So whenever you're making chlorophylls, those intermediates have to be bound in some protein that can do something with that light energy. Otherwise, the plant would burn itself up from the inside out while it's making chlorophylls. Right? So it's a lot of really interesting chemistry that goes on. We think about enzymes as working on substrates that are floating around free in the cytoplasm or in the stroma. This is a completely different picture here.
Would it be that ABA somehow triggers a change in the structure of that protein that allows them to get in there?
Oh, certainly that has to happen, but ABA doesn't trigger that. Because remember, ABA is the product. It's what's being made, right? Something is going to have to trigger that to turn on enzymes that are going to convert these carotenoids into ABA. But I just wanted to bring to your attention that this is a totally different type of enzyme chemistry than what we normally think about, because it's happening with substrates that are embedded in the middle of very hydrophobic proteins. Okay, I'm not going to go into the details of all the reactions and the mutants that have been identified there, but I do want to say something briefly about the metabolism of abscisic acid. It fits into the same categories that we've been talking about with other hormones. There are reversible formations of conjugates. In this case, it's adding a glucose residue makes it into an inactive compound, and that glucose can be removed and reactivate the compound. The role that ABA conjugates play in physiology is not very well understood. So we'll talk in just a couple of minutes about ABA that's synthesized in the leaves is transported down to the roots, and then it goes back up to the leaves under drought stress conditions. What happens to the ABA in the roots? Is it conjugated? There's not very good evidence to support that. ABA can also be broken down locally, and that can have dramatic effects on the ABA concentration as well. Okay, let's think for a second about ABA transport. This is a story you've seen already, but it's worth repeating. If we think about ABA as being a weak acid, right? So it can pick up a proton and form the protonated form. And if this is a low pH region, high concentration of protons, it's going to favor the protonation over the deprotonation reaction. So we would expect this, the protonated form, to dominate in the low pH regions. But because of the structure of ABA, it's a pretty hydrophobic molecule. So when it's not charged, when it's in its neutral form, ABA can reversibly move back and forth across membranes. You don't need a transporter to get ABA into and out of cells. Once it gets in the cell, typically the pH in the cell is higher, more basic. So that's going to favor the deprotonation reaction. So in the cells, the charged deprotonated form will dominate, and this cannot cross a membrane. So like we saw with auxins, the distribution, the cellular distribution of ABA is going to be, at least in part, governed by pH. So under normal conditions, in a leaf cell, in the daytime, where are you going to find most of the ABA? It's not in the book. Don't search your mind for something you read. Think about it. Where would most of the ABA be located based on the information we've provided here? High pH or low pH? High pH. Where's the highest pH going to be in a leaf cell in the light? Why would a lot of sugar change the pH? Anna?
You know what, that for the water gets split?
Why was that? Doesn't that release protons?
Right. Oh, yeah.
Come on, guys.
I think it's in the stroma of the chloroplast.
Why do you say that?
Because the hydrogen just pump hydrogen outside of chloroplast, so that's true.
Almost right. Remember that photosynthetic electron transport pumps protons from the stroma into the lumen of the thylakoids. So where's the highest pH going to be? In the stroma, right? So in the light, in normal leaf cells, where's most of the ABA going to be? In the stroma, right? In the place in the cell where the pH is the lowest. And that's the correct answer.
Is the ionized form inactive?
Is it inactive? No, it's not inactive. It just can't cross membranes.
Oh, okay.
That's a good question. Is this form an active form, or is the protonated form an active form? As far as we know, they both have activity. Okay, so we don't need to have oxygen, or we don't need to have ABA transporters. It gets across membranes just fine on its own. But the distribution of ABA, like auxin, is going to be influenced by differences in pH across membranes. It will tend to accumulate in regions with higher pH because it favors the formation of the charged form, and the charged form then can't get back across the membrane and gets trapped. Okay. This tells us what's going on at the cellular level. What's going on at the whole plant level? How does ABA get around in the whole plant? It's mostly in the vascular tissue. Although what we hear about is ABA moving in the xylem from the roots to the leaves under conditions of water stress. Under normal conditions, ABA is moving in the opposite direction. The majority of the ABA in plants is synthesized in the leaves and transported to the roots in the phloem. And what happens to that ABA in the roots is an interesting question. So some very interesting labeling studies have been done. You grow plants in the presence of C14CO2, and the ABA that is synthesized in the leaves has that radioactive label and moves down to the roots. Then if you drought stress the plants, that same ABA with the radioactive label in it moves from the roots back up to the leaves and signals all the stomatal closing and things like that. So it's pretty clear that the ABA supply in the roots is to at least some extent dependent upon ABA that's synthesized in the leaves and moved down to the roots in the phloem under sort of normal conditions. Right? So the roots are also capable of synthesizing ABA, but plants' responses to drought stress are greatly decreased if you inhibit ABA synthesis in the leaves. And that happens for two different reasons, and we'll see why in just a minute. Okay, so let's think about what's going on in a leaf when the roots are water stressed. So one of the things we know is going to happen from strictly water potential picture is if you decrease the amount of water that's available in the roots, you make the soil drier, so its water potential becomes more negative. That is reflected by decreases in water potential in the leaf, right? Water loss, increased solute concentrations, all of that leads to more negative water potentials in the leaf, right? Stomatal closure, right? So if we look at the closing of the stomates, not surprisingly, as water stress continues, the stomates remain closed. And ABA content pretty much parallels that. How much is ABA, what role is ABA playing in closing those stomates? So the book doesn't do a very good job of discussing this. It's relatively complicated. There's several things that are going on. Yeah, so let's save talking about the signal transduction pathway associated with ABA and stomatal closing till the end. That last diagram in the chapter, a number of you asked questions about that. I mean, that's basically a worthless diagram, right? It tells you that there's ABA receptors, it tells you that there's calcium, and it tells you that there's potassium and chloride channels, and that there's something interesting going on there, right? Well, all that expresses is how little we know about what's really happening in the ABA signaling pathways. But we want to think about stomatal closure, drought-induced stomatal closure. And the book talks about two phases of this, sort of the immediate phase and then the longer-term phase. And the difference between immediate phase and longer-term phase is, sort of minutes to hours versus days. Okay, so let's talk about what's happening first in this immediate phase. There's two things going on. If you lower the water potential in the roots, if the soil becomes dry, or if you added salt to the soil, how long will it take before the leaves sense that change in water potential? Let me rephrase the question. What connects the water potential in the soil to the water potential in the leaves? Yeah, right, transported in the xylem, right? So as soon as it's harder to take up water in the soil, it's going to be almost immediately harder to replace water that's being lost in the leaves. So we expect that connection to happen pretty quickly. So if the water potential drops in the soil, the water potential in leaves, or at least in the apoplast of the leaves, will also drop, right? If the water potential in the apoplast of the leaves drops, what happens to the water potential of the mesophyll cells in the leaf? It also drops, right? So there is a hydroactive component to stomatal closure that has nothing more than the lowering of the water potential in cells in the leaf due to the lowering of the water potential in the xylem and in the roots. Right? So this is an immediate effect. It has nothing to do with anything more than changes in the water potential induced by changes in the water potential in the soil. So one aspect of this immediate phase is this hydroactive component. But there's also what's referred to as a hydropassive component. And this is local ABA synthesis in the leaves. So these immediate effects are happening on timescales of minutes to hours. And if we consider the long-term effects, we have exactly the same components. There's a hydroactive component to this. If the soil water potential remains low, then that will continue to have its effect on the water potential in leaves. The hydropassive component in the long term is not due to ABA that's produced locally. It's due to ABA that is being translocated from the roots up to the leaves. So in this hydropassive component, in the long term, this is ABA transport from the root. And we'll see this whole plant level redistribution of ABA has other effects than just closing the stomates. Okay, so the role that ABA transport is playing in this is primarily happening in this long-term effect in ABA transport from the roots. In the shorter term, ABA transport is going on, but it's basically transport that's happening at the cellular level. Now, there's one other component to this that the book mentions. It's an interesting one, and it's maybe a little bit complicated to think about, and that is, it turns out that when the roots become water stressed, the pH of xylem sap becomes more basic. So under well-watered conditions, the pH of xylem sap is around 6.3, and under water stress, it becomes just slightly basic, around 7.2. How is this going to affect the distribution of ABA within the leaf? Well, remember, we talked about the fact that ABA tends to accumulate in regions where the pH is lower. So if we're only going to consider ABA in cells versus out of cells, under well-watered conditions, the ABA would be favored to be in the cytoplasm or inside the cell because the pH of the apoplast is more acidic. But under water stress conditions, if the pH of the apoplast becomes more basic, you're shifting the equilibrium of ABA distribution from being primarily in the cells to having more of it in the apoplast. So how does this affect stomatal regulation? So let's say that you've got 100 units of ABA that are present in the leaf. Under well-watered conditions, where's most of that ABA going to be located? inside the cells. And what fraction of the cells in the leaf do the guard cells represent? Pretty small fraction, right? So if all these mesophyll cells are taking up ABA, then the guard cells aren't going to see very much of it. So the effect on the guard cells is relatively small. But as the pH of the xylem sap becomes more basic, more of that ABA stays in the apoplast, which means it's more available to get to the guard cells. So there's a third component down here in the long-term effects, and that's a pH-dependent redistribution of ABA due to changes in the pH of the xylem sap. So all of these things are working together to control stomatal aperture in the short term and in the long term. So there's common components. There's both hydroactive and hydropassive components to both the short term and the long terms. The difference is short term is local ABA, long term is transport from the roots. And there's also this pH dependence that's affecting the ABA distribution in the leaves as well. Okay? So this physiology should be relatively straightforward. Should be able to understand this pretty well. Grace, you have a question?
You said something about the pH-dependent redistribution of ABA in the xylem sap, as opposed to, were you just talking about the apoplast?
I'm just talking about the apoplast versus the symplast in the root. So it's an interesting question. Why does the pH of the xylem sap change as the soil dries down? And I don't know the answer to that. I'll be honest, I haven't looked for the answer to it, but I don't know what it is. But the consequences are important in terms of keeping the stomates closed under water-limiting conditions in the leaves in the long term. Okay? All right. Let's delicately step into thinking about signal transduction pathways in ABA. So one of the key things that I mentioned about ABA can't spell. Unlike all the other hormones that we've talked about, there's no ABA receptors that have been definitively identified. The book talks about a number of different possibilities, and they're interesting possibilities. I want to talk about two aspects of this. Let's see, I can't remember if I have a slide on this. Nope, I don't. I want to talk about two different aspects of the receptors. One of them is the possibility that the CHLH protein plays a role in ABA signaling. So it's pretty clear that this protein, first of all, what is this protein? This is a subunit of a rather large protein complex that's involved in magnesium chelation. The magnesium chelatase. So in chlorophyll biosynthesis, remember the middle of the porphyrin ring in chlorophyll has got a magnesium in it, in contrast to iron that's in the presence of a heme ring. So there's an enzyme that sticks the magnesium into chlorophyll. And actually, it is at this step where the intermediates in chlorophyll biosynthesis, before this step, they're floating around free in the stroma. After this step, they're all bound to proteins, because after this step, the chlorophyll absorbs light very effectively, or the chlorophyll precursor absorbs light very effectively. So the key thing that this protein is doing is that it acts as an enzyme associated with chlorophyll biosynthesis. But it plays another, recently identified, another very important role, and that is signaling between the chloroplast and the nucleus. We haven't really talked about this, but for example, when we think about the proteins that function in the chloroplast, let's think about the photosynthetic proteins, reaction centers, key things that happen that are required for photosynthesis. reaction centers have, I can't remember, photosystem 1 has 19 different proteins and photosystem 2 has got 20-something of them, right? Some of those proteins are encoded in the chloroplast. Most of them are encoded in the nucleus. Rubisco. Large subunit of Rubisco is encoded in the chloroplast. Small subunit of Rubisco is encoded in the nucleus, right? There has to be coordination between the nuclear and chloroplast genomes in making sure that you don't have too much of one and too little of another. And it turns out that this CHLH protein also plays a critical role in regulating that chloroplast nuclear transcriptional regulation. And it makes some sense because this also plays a key role in going from precursors that are not dedicated to chlorophyll to precursors that are dedicated to chlorophyll. And most of what the proteins are doing in the chloroplast have something to do with photosynthesis. Okay? So the fact that this is playing a key role in this signaling is perhaps not so surprising. We don't really understand this very well. In fact, we have 4 graduate students in plant biology whose PhD project is related to this signaling process, the role that this protein is playing in it. Okay, so it's something that we don't really understand very much other than that this protein plays a key role. And one of the things that we know at this point is that the part of the magnesium chelatase that this protein plays in the enzyme catalysis is a different domain of the protein than the part that plays its role in signaling. So in other words, it's like it had 2 different proteins that got joined together into one. One part of the protein does one thing and one part of the protein does another thing. Well, the picture just got more complicated because the other thing is this is ABA binding. There's a domain in there, in this same protein, different domain than these 2, that is capable of binding ABA. That's basically all we know at this point. But the fact that this protein plays a critical role in signaling associated with control of transcription in the chloroplast and the nucleus, it certainly makes sense that if ABA binds to this protein, that it could be involved in a signal transduction pathway. What the link is between these 2, we don't know. Okay? So this is one example of we haven't really identified any definitive receptors yet, but this would make sense if it worked this way. Okay? Another one that the book talks about from a different perspective. There's signaling associated with ABA that involves 3, what's the right term? Second messengers, non-protein components of the signal transduction pathway. One of these is diacylglycerol. It's a breakdown product of lipids in the membranes of cells. Another one is IP3, inositol triphosphate. We've seen that before. You've seen it before. You've seen both of these before. And the other one is calcium ions. So, any of you who took introductory biology from me, none of you did, but if you did, this information should ring a bell right in your head. It should say G protein linked receptors. Because the signal transduction pathways that use IP3, diacylglycerol, and calcium is signaling molecules in the signal transduction pathway. The most common ones are G protein linked receptors. There is no G protein linked receptor that's been identified in plants. But the fact that there are these intermediates play a role in ABA signaling, and the fact that ABA is made in many other organisms beside plants, and in those organisms use a G protein linked receptor pathway, signaling pathway, suggests that there may also be one in plants. You would think that this would be relatively easy to identify. So we have, the genome of half a dozen different plants sequenced. We know what the amino acid sequence of G protein linked receptors are in lots of other types of organisms. Why can't we look for homologous sequences in Arabidopsis or in rice or things and find them there? Well, you think that we could, but it hasn't worked. So there's interesting questions here. Are there really G protein linked receptors in plants? Certainly the components of the signal transduction pathway would suggest so. Because if you inhibit, for example, the phospholipase that's involved in formation of these two products in the signal transduction pathway, you inhibit ABA responses, or you inhibit some ABA responses in plants. Okay? So there's lots of circumstantial evidence for very interesting things going on with the ABA receptors. Different than what we've seen before. So, as the book talks about, there's evidence for at least 3 completely different types of ABA receptors. We haven't seen that for any of the other hormones. All the other hormones have a common pool of hormone receptors that then branch off in terms of what the signal transduction pathways do. Okay, so some pretty bizarre stuff going on here. Okay, let's talk about ABA in seed development, and that may be the last thing that we get to today. So any of you taken any seed physiology classes? Nope, okay. So when we think about what happens in a seed, we've talked about the first aspect of seed development. This is basically embryogenesis. So for any type of seed, the seed development starts off with the formation and growth of the embryo, the sorts of things that we talked about several lectures ago. The second phase involves the production of basically storage products. Seeds contain varying amounts of products, whether they're stored in the endosperm or whether they're stored in the cotyledons, doesn't really matter. Those are compounds that when the seed germinates, when that embryo starts to grow, will supply that growing embryo with the carbon and the nitrogen and everything it needs until the seedling gets above the surface of the soil and can start doing photosynthesis, right? So the formation of these storage products is an obligate part of this seed development. And the last phase is drying down. So seeds typically lose 95 to 98% of their water in this drying down phase. And what this also includes is sort of metabolic stasis. Suspended animation, basically, for the embryo. It's dried, the metabolism is basically stopped. But when water comes back, the drying down of the tissues has been done in such a way that the ability to continue to grow is still there. Okay, so normally what happens after this, the seed sits for some period of time, and then it picks up water. And the seed germinates. Well, it germinates unless it's dormant. And then dormancy does something to prevent the seed from germinating, even though water is present. Right? So there's several components of this seed development and regulation of seed growth that ABA play roles in. If we look at the distribution of ABA as we go down through these various steps here. So we're going to make a graph here where ABA content increases in this direction. ABA is relatively low at the beginning, rises rapidly sort of in the middle of phase 1, and then declines very slowly until you get down here to germination, then the ABA level gets low again. It turns out the ABA is playing a number of different roles here. At this point, ABA plays an important role in turning on the synthesis of compounds that are going to be used as storage products. Okay, so ABA is regulating the creation of these storage reserves in the seeds. So ABA is playing an important role here. ABA also plays a critical role in the drying process, not so much in inducing the drying, but if you take a normal embryo and dry it down, if you take an embryo at the end of embryogenesis and dry it down and then give it moisture back again, the embryo's dead. There's something happening between embryogenesis and the drying process that is protecting that embryo during the dry state. Right? And we've talked about these sorts of compounds. Put a little arrow over here, the formation of compatible solutes. Compounds that can protect membranes, proteins that are normally interacting with water to function are produced during the second stage of seed development, so that when the seed dries down, it's got everything built into it. There's a whole group of genes that are turned on in this second phase. They're referred to as the LEA genes, late embryogenesis, abundant. So the LEA genes are a group of genes that are involved in synthesizing these compatible solutes, so that when the seed dries down, when the embryo goes into metabolic stasis, the structure of the membranes and the proteins are preserved so that when water comes back again, the seed can actually grow. Okay? So these are ABA effects that are affecting the seed in terms of controlling gene expression. But ABA plays another really important role here, one we've already mentioned, the role that ABA plays in dormancy. Seeds that aren't dormant, you give them water, they start growing right away. Seeds that are dormant, their ability to turn on metabolic function is limited by abscisic acid. ABA is a compound that maintains dormancy. Get rid of the ABA, or as we'll see, increase the amount of gibberellic acid there, and that dormancy can be broken. When does this dormancy start? When does this dormancy start?
Isn't that as soon as the cell dries, like the seed dries, it's technically like dormant until the
When does dormancy start? It didn't start at all. Okay, but so in other words, these embryos started growing. Where are those embryos in this process? They're before drying, right? These corn kernels haven't dried down yet, right? So that dormancy is being imposed early on, sort of in this sort of region of seed development, as being maintained until the right environmental conditions trigger the ending of dormancy. That ending of dormancy involves primarily decreasing the amount of ABA and increasing the amount of gibberellic acid, right? So seed dormancy is imposed on the embryo early in when embryogenesis is done, early in this phase of the storage of products there. Because you can see that the corn kernels are reasonably filled out here, but they're definitely not dried down, right? So one important thing that the book emphasizes, and a couple of you asked questions about this, the ABA that is controlling these processes, the formation of products and the formation of the compatible solutes, the late embryogenesis, that ABA is produced in the embryo. It's being controlled within the embryo. This ABA that is inducing and maintaining dormancy is, at least initially, produced by maternal tissues. So the mutations that affect vivipary are mutations that affect ABA production in maternal tissues, not in the embryo itself. So 2 different sources of ABA, maternal, embryonic, 2 different processes they're controlling. Embryo, the production of storage products from the embryo and the production of compatible solutes that protect the embryo when it dries down. That's embryo-produced ABA. But the imposition of dormancy is done by ABA produced by maternal tissues. And you should be able to say, how does it tell what ABA is which? Right? This ABA and this ABA are the same thing. What determines whether that ABA leads to the formation of these compounds or whether that ABA induces dormancy? Now, we're not going to answer it now. That's something you should be able to answer, for example, on an exam question. Okay. We'll finish off next time. I just want to talk a little bit about ABA and blood dormancy. It's pretty much the same story. But I also want to talk about the whole plant distribution of ABA. One of the things that we need to think about, one of the things that you should expect to happen. What's going to happen to shoot growth and root growth in plants that are water stressed? What would you expect would happen to shoot growth and root growth in plants that are water stressed? Patrick, what would you think would happen there?
How do you expect it to slow?
Both of them to slow?
Well, root growth to go farther to find more water than shoot growth.
Good. Yeah, that's basically it, right? I mean, physiologically, you would expect the plant would reallocate resources away from shoot growth and toward root growth if the plant is water stressed. And that's exactly what happens. And you should be able to tell me what dictates that. How is that controlled? How does water stress control shoot growth versus root growth based on the things we talked about? We'll come back and talk about that at the beginning of the next lecture. And then we'll do grass and steroids.